We are going to start with the chapter number 16, excretory products and their elimination, NCERT reading. Let's start. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water and ions like Na positive, potassium, Cl negative, phosphate, etc. either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter you will learn the mechanisms of elimination of these substances with a special emphasis on common nitrogenous wastes, that is ammonia, urea and uric acid. They are major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form, yes, and requires a large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia is a monotelism. Many bony fishes, aquatic amphibians and aquatic insects. Bony fishes, aquatic amphibians and aquatic insects, they are monotelic in nature. Ammonia, as it is readily soluble in water, is generally excreted by diffusion across the body surfaces or through gills surfaces in fishes as ammonium ions. Kidneys do not play any significant role in its removal. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous wastes like urea and uric acid for conservation of water. Mammals and many terrestrial amphibians and marine fishes mainly excrete urea and are ureotelic. Mammals, many terrestrial amphibians, marine fishes. Here is bony fishes, yes, aquatic amphibians, aquatic insects, mammals, many terrestrial amphibians and marine fishes. They mainly excrete urea and are called ureotelic. And ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolality. Reptiles, birds, land snails, insects and tadpoles, they excrete nitrogenous waste as uric acid in the form of a pellet or paste. With the minimum loss of water, they are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures in most of the invertebrates. These structures are simple tabular forms, whereas vertebrates have complex tubular organs called kidneys. Some of these structures are mentioned here. Protonephridia or flame cells are excretory structures in platyhelminthes, flatworms, etc., planaria, rotifers, some annelids, and the cephalocaudate amphibians. Yes, protonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are the tubular excretory structures of earthworm and other annelids. Nephridia helps to remove nitrogenous waste and maintain a fluid and ionic balance. Malphigian tubules are the excretory structures of most of the insects, including cockroaches. Malphigian tubules helps in the removal of nitrogenous wastes and osmoregulation. Antennal glands or green glands perform excretory function in crustaceans like prawns. Crustaceans like prawns, they have antennal glands or green glands. As kingdom, arthropoda is divided into three: crustaceans, insecta and arachnoids. Yes, crustaceans contain what? Antennal glands or green glands. Insecta contain Malphigian tubules and arachnoids contain coxal glands. Yes, for excretion. Human excretory system. Look, adrenal gland कहाँ है? किडनी के ऊपर. Renal artery, renal vein, yes, kidney, dorsal aorta, ureter, cortex, medulla, pelvis, urinary bladder and urethra. ठीक है. In humans, excretory system consists of pair of kidneys, one pair of ureter, a urinary bladder and a urethra. Kidneys are reddish brown bean-shaped structure situated between the levels of last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width and 2 to 3 cm in thickness, with an average weight of 120 to 170 gram. Towards the center of the inner concave surface of the kidney is a notch called hilum, through which ureter blood vessels and nerves enter. Inner to hilum is a broad funnel-shaped space called renal pelvis with projection called calyxes. The outer left kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and inner medulla. Medulla is divided into few conical masses. Yes, projecting into calyxes. The cortex extends in between medullary pyramids as the renal columns and called columns of Burton. Each kidney has a nearly 1 million complex tubular structures called nephrons, which are functional units. Each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is a tough capillaries formed by efferent arteriole, a fine branch of renal artery. Blood from glomerulus is carried away by efferent arteriole, and the renal tubule begins with a double-walled cup-like structure called Bowman's capsule, which contains podocyte on the inner membrane, which encloses the glomerulus. Glomerulus along with the Bowman's capsule is called Malpighian body or renal tubule renal corpuscle. The tubule continues further to form a highly coiled network called PCT, proximal convoluted tubule. Yes. Now ahead, a hairpin shaped Hanel's loop is the next part of tubule, which has a descending and ascending limb. This makes the urine more concentrated. The ascending limb continues as another high coiled tubular region called distant convoluted tubule, DCT. The DCTs of many nephrons opens into a straight tube called collecting duct, many of which coverage and open into the renal pelvis through medullary pyramids and the calyxes. The Malpighian corpuscle, PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas a loop of Hanel's dips into the medulla. In majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons, they are called cortical nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called the peritubular capillaries. A minute vesicles of this network run parallel to Hanel's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. Moving ahead, urine formation. Urine formation involves three main processes, namely glomerular filtration, which is a non-selective process, reabsorption and secretion that takes place in different parts of the nephrons. The first step in urine formation is filtration of blood, which is carried out by glomerulus and is called glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidney per minute, which constitutes roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, that is the endothelium of glomerular blood vessel, the epithelium of Bowman's capsule, and the basement membrane between the two layers. The epithelial cells of Bowman's capsule, called podocytes, arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the protein pass into the lumen. All the constituents of plasma except proteins, they pass onto the lumen of Bowman's capsule. Therefore, it is considered as the process of ultrafiltration. And moving ahead is the amount of filtrate formed by kidneys per minute is glomerular filtration rate. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liters per day. The kidney have built in a mechanism for regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus, JGA. JGA is a sensitive region formed by cellular modification in the distal convoluted tubule and the afferent arteriole at the location of the contact. A fall in GFR can activate the JG cell to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to the normal. A comparison of the volume of the filtrate formed per day with that of urine release suggests that 99% of the filtrate has to be reabsorbed by the renal tubules. This process is reabsorption. The tubular epithelial cells in different segments of nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acids, and positive in the filtrate are reabsorbed actively, whereas nitrogenous wastes are absorbed by passive transport. Reabsorption of water also occurs passively in initial segments of the nephrons. During urine formation, the tubular cells secrete H positive, K positive ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and acid-base balance of body fluids. Function of the tubules, PCT. This is lined by simple cuboidal, brush-bordered epithelium, which increases the surface area for reabsorption nearly all of the essential nutrients, and 70 to 80% of electrolytes and waters are reabsorbed by the segment. PCT also helps to maintain the pH and ionic balance of body fluids by selective secretion of H positive ammonia into the filtrate and absorption of bicarbonate from it. Hanel's loop. Reabsorption is minimum in its ascending limb. However, this region plays a significant role in maintenance of high osmolarity of medullary interstitial fluid. The descending limb of loop of Henle is permeable to water but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrate pass upward, it gets diluted due to the passage of electrolytes to the medullary fluid. DCT, distal convoluted tubule. Conditional reabsorption of Na positive and water takes place in this segment. DCT is also capable of reabsorption of bicarbonate and selective secretion of hydrogen and potassium and ammonia to maintain the pH and sodium potassium balance in blood. Now moving ahead is this vasa collecting duct. This long duct extends from cortex of the kidney to inner parts of medulla, and large amount of water could be reabsorbed from this region to produce a concentrated urine. This segment allows the passage of small amounts of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in the maintenance of pH and ionic balance of blood by selective secretion of H positive and K positive ions. Now the mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta play a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite direction. This forms a countercurrent. The flow of blood through the two limbs of vasa recta is again a countercurrent pattern. The proximity between Henle's loop and vasa recta as well as countercurrent in them help in maintaining an increasing osmolarity towards inner medullary interstitium. That is from 302 in cortex to 1200 in inner medulla. This gradient is mainly caused by NaCl and urea. NaCl is transported by ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. NaCl is returned to interstitium by ascending portion of vasa recta. Similarly, small amounts of urea enter the thin segment of ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. The above described transport of substance facilitated by special arrangement of Henle loop and vasa recta is called countercurrent mechanism. This mechanism helps to maintain a concentration gradient. This is how osmolarity of this is. In cortex it is 300 and medulla it is 1200. In the medullary interstitium, presence of such interstitial gradient helps in an easy passage of water from collecting tubule, thereby concentrating the filtrate. Human kidney can produce urine nearly 4 times concentrated than the initial filtrate formed. Regulation of kidney functions. Functioning of kidney is efficiently monitored and regulated by hormonal feedback mechanisms involving in hypothalamus, JGA, and to a certain extent the heart. Osmoreceptors in the bodies are activated by changes in blood volume, body fluid volume, and ionic concentration. And excessive loss of fluid from the body can activate these receptors, which stimulate the hypothalamus to release antidiuretic hormone or vasopressin from the neurohypophysis, which is posterior pituitary. And ADH facilitates water reabsorption from later parts of the tubule, thereby preventing diuresis. An increase in body fluid volume can switch off the receptors and suppress the ADH release. To complete the feedback, ADH can also affect the kidney function by its constrictory effects on blood vessel. This causes an increase in blood pressure. An increase in blood pressure can cause increase in the glomerular blood flow, that is GFR again. The JGA plays a complex regulatory role. A fall in GFR, glomerular blood pressure, can activate JG cell to release renin. Renin converts angiotensinogen in the blood to angiotensinogen 1, further to angiotensin 2. Angiotensin 2 being a powerful vasoconstrictor increases the glomerular blood pressure and thereby GFR. Angiotensin 2 also activates the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of sodium and water from the distal parts of tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as renin-angiotensin mechanism. An increase in blood flow to the artery of the heart can cause the release of arterial natriuretic factor. ANF can cause vasodilation of blood vessel, thereby decrease the blood pressure. ANF mechanism therefore acts as a check on the renin-angiotensinogen mechanism. Micturition. Urine formed by nephrons will ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system. The signal is initiated by stretching of urinary bladder as it gets filled with urine. In response, the stretch receptors on the walls of the bladder send signals to the CNS. The CNS passes on motor messages to initiate the contraction of smooth muscles of bladder and simultaneous relaxation of the urethral sphincter, causing the release of urine. The release of urine. This process is known as micturition. And the neural mechanism causing it is called micturition reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. Urine formed is a yellow colored watery fluid which is slightly acidic. pH 6 has a characteristic odor. On an average 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristics of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of kidney. For example, presence of glucose, glycosuria and ketone bodies, ketonuria and urine are indicative of diabetes mellitus. Now, the role of other organs in excretion. Other than kidney, lungs, liver and skin. Other than the kidneys, lungs, liver and skin also helps in elimination. Our lungs remove large amount of carbon dioxide and also significant quantities of water every day. Liver, the largest gland in our body, secretes bile containing substances like bilirubin, biliverdin, cholesterol degradated, steroid hormones, vitamins and drugs. Most of these substances ultimately pass out along with the digestive waste. The sweat and sebaceous gland in the skin can eliminate certain substances through the secretion. Sweat produced by sweat glands is a watery fluid containing NaCl and small amounts of urea, lactic acid, etc. The primary function of sweat is to facilitate a cooling effect on the body surface. It also helps in the removal of some of the waste as mentioned above. Sebaceous gland eliminates certain substances like sterols, hydrocarbon and waxes through sebum. This secretion provides a protective oily covering for the skin. Do you know that small amounts of nitrogenous waste could also be eliminated through saliva too? Disorders of excretory system. Malfunctioning of kidney can lead to accumulation of urea in the blood condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from a convenient artery is pumped into the dialyzing unit called artificial kidney. Blood drained from the convenient artery is pumped into dialyzing unit after adding an anticoagulant like heparin. The units contain a coiled cellophane tube surrounded by a fluid, which is a dialyzing fluid having the same composition as that of plasma except nitrogenous waste. The porous cellophane membrane of the tube allows the passage of molecules based on concentration gradient. As nitrogenous wastes are absent in dialyzing fluid, these substances freely move out, thereby clearing the blood. Cleared blood is pumped back to the body through a vein after adding antiheparin to it. This method is a boon for thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in the correction of acute renal failures. There is a kidney failure. A functioning kidney is used in transplantation from a donor, preferably a close relative, to minimize its chances of rejection by the immune system of the host. Modern clinical procedures have increased the success rate of such a complicated technique. Renal calculi, stone or insoluble mass of crystallized salt, oxalates, etc., formed within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney.